I'm going to present my master thesis based in assessing the presence of microplastics in marine macrophytes for the possible use in bioremediation. Plastic pollution has increased in last decades with 135 million tons. Plastic debris can reach the environment through river basins, inland factories, and offshore activities like fisheries. Microplastics can be a toxic threat due to the additives that are released in the ocean, and the bioavailability of these particles makes them part of the diet of being reported their ingestion in invertebrates and vertebrates. In contrast, studies about microplastics accumulation have been limited for marine macrophytes. These autotrophic organisms represent essential ecosystem services because they offer sediment stabilization, they provide habitat, protection, and food for a lot of organisms. Their morphology allows the retention of suspended particles, the reduction of water flow, and studies have demonstrated their capacity to uptake pollutants and absorb nutrients, making them suitable to be used in bioremediation, which is an eco-friendly and cost-effective technology that uses living organisms to eliminate environmental pollutants. When this technique uses green plants for this process, this is called phytoremediation, and higher eukaryotes break down pollutants by phytoextraction when they introduce the pollutant into structures and produce degradation, and also by rhizofiltration when they break down the pollutants throughout the root system. These two processes can be used for microplastics bioremediation, even though this has been less investigated. Therefore, the aims of my study were to explore marine macrophytes from published results that could be suitable for microplastics bioremediation by evaluating the capacity of marine macrophytes to retain microplastics, analyzing biological and ecological aspects for the selection of potential candidates for microplastics bioremediation, and assessing qualitatively the best suited species for microplastics bioremediation. For the literature research, articles and publications were selected via Scopus and Google Scholar databases according to two criteria. First, publications were based in the presence of microplastics in at least one macrophyte structure. And second, studies were focused in the presence of microplastics in sediments that cover the locations where criterion 1 was met. When the data was not found for the same place, another studies were searched in the locations close by, and these were included as reference. Then, to show the efficacy of a species to uptake a pollutant from the environment, it was necessary to measure the bioconcentration. And in this case, it was by dividing the number of microplastics per species of marine macrophytes by the number of microplastics present in sediment. So the high values will indicate a greater potential of the species to be accumulated pollutant. A series of features had to be considered for a macrophyte to be suitable for microplastics bioremediation, and these were: first, the species needed to present high and fast growth rates in order to have a strong bioremediation potential. Two, species needed to be employed only inside their native range, or preferably they needed to be widely distributed to prevent invasions and protect biodiversity. Three, species needed to exhibit high tolerance to environmental conditions like temperature and salinity. Four, species should have efficient and diverse mechanisms for microplastics retention in their structures. And five, MP's retention rates needed to be high and adequate to ensure particles will not end back in the environment. For the assessment of species suitability for bioremediation, the 5 features that I mentioned before were evaluated for each species. I gave a rank from 1 to 16 for each species, being 1 not suitable and 16 most suitable. And then the scores were added for each feature, and the species with the highest values were selected as suitable candidates for microplastics bioremediation. As results, 227 articles were retrieved from 2006 until early 2020, and 14 studies met the 2 criteria for selection. And as you can see in the figure, The increasing number of publications reflects the interest of scientific community in microplastics investigation, and also shows the very recent interest in microplastics content in macroalgae and seagrasses, with publications only in the last 5 years. A total of 16 species of marine macrophytes were mentioned for the presence of microplastics. And according to the taxonomic classification, they were divided in macroalgae with 11 species and seagrasses with 5 species. Regarding the bioconcentration, for macroalgae, genus Pyropia and species Ulva prolifera and Endocladia muricata, and seagrasses Cymodocea rotundata, Cymodocea cerulata, and Thalassium prichi had the highest values for this factor. For the mechanisms for retention of microplastics in these macrophytes, for macroalgae, we have that the presence of biofilm and polysaccharide mucus layer allows the microplastics to be absorbed in their structures, like, for example, in blades, in some species in air sac and the stipe. Also, the morphology of macroalgae allowed these microplastics to be either wrapped, twinned, or embedded in the structures. For seagrasses, biofilm and epibiontic organisms help microplastics to be trapped in their structures, like, for example, leaves. And the dense branching of these angiosperms reduces water flow, causing microplastics that are suspended in water column to be sinked in sediment, and thus producing microplastics accumulation. So to carry out the assessment properly, some biological and ecological characteristics were searched in literature to give the correct score for each feature evaluated. This table shows how different factors were contributed for species to be either discarded or have high or low scores in the evaluation. I'm just going to mention some examples because the table had a lot of information. And it was very big for the 16 species, and it was not possible to show it completely here in the presentation. For example, for Undaria pinnatifida, it was discarded for the assessment because, as you can see here, the fact that it's an alien and invasive species doesn't make it choosable for bioremediation. Also, for species like Endocladia muricata, in some species like this one, the lack of information, like, for example, environmental conditions, like you can see here, made this species to have low scores in some features like that one. And for example, for Ulva prolifera, the information found in literature was a lot. So for this one was one of the only species that I could find specific growth rates, like you can see here, per day. And also it was widely distributed, and the high tolerance, as you can see, in salinity and temperature was very, very high. So this made this species to have high scores for the assessment. For seagrasses, in species like Cymodocea cerulata, it had narrow distribution and low tolerance to environmental conditions. So for these features, this species had low scores. But for Zostera marina, it was all the way around. The wide distribution of this species, and it was one of the most tolerant ones per seagrasses, made it to have high scores for the assessment. So according to the assessment, the potential candidates for microplastics bioremediation were for macroalgae, Ulva prolifera with a score of 74 points, and seagrass Zostera marina with a score of 60 points. Ulva prolifera is a bloom-forming algae, and studies have shown that microplastics do not cause any structural damage or interrupt any biological processes. So it makes this species a good ally for bioremediation. And for instance, it can be used for microplastics, to treat microplastics for contaminated effluents offshore, like commercial fishing, mariculture, and aquaculture. In the case of seagrass Zostera marina, besides the mechanisms to trap microplastics, in situ studies have demonstrated its bioremediation capacity to remove PCBs from sediment. And also, it can be used to trap microplastics from nearshore effluents due to the high tolerance to environmental conditions. These two species could be used to retain microplastics in wastewater treatment plants, only taking into account the prevention of propagule dispersion and the adequate disposal treatments. As new research approaches, an alternative for an optimal performance of marine macrophytes is to explore synergistic relationship with a specific microbiota to enhance proficiency in bioremediation. Also, further studies are fundamental to corroborate in situ the experimental studies mentioned in here to demonstrate the potential suitability of marine macrophytes in bioremediation. And also, the applications of these and other technologies are key to prevent microplastics to enter in the oceans and become a threat to marine flora and fauna. As conclusions, we have that macroalga, Ulva prolifera, and seagrass, Zostera marina, were selected as potential candidates for microplastics bioremediation due to the optimal and efficient biological and ecological features they have. And if confirmed from further studies in situ and experiments, species could be proposed for their use in bioremediation in different locations, like wastewater treatment plants, coastal and offshore effluents. I want to thank my promoter, Eva Garcia, and my supervisor, Alba Ardura, for always giving me their support through all the process. Also, I want to give a special thanks to PhD student Paula Macia for always being open to help me. I want to thank my mom, my dad, and my sisters for always giving me their support despite the distance. And also to my friends back home and my friends, my IMBRSea friends, for always being there to cheer me up. Any questions?